Welcome back, everyone. In this presentation, we're going to continue our discussion on basic biochemistry. We'll talk about nucleic acids and we'll talk about proteins. And then we'll end the discussion talking about compartmentalization, a very extremely important concept when we start talking about physiologic processes. So, nucleic acids consist of the following. They consist of one or more 5-carbon sugars, one or more phosphate groups, and a nitrogenous base. When we think about nucleic acids, the basic unit of a nucleic acid is something called a nucleotide. And a nucleotide consists of one of each of the 5-carbon sugar, one phosphate group, and one nitrogenous base. The nitrogenous bases, there's actually several of them. And these nitrogenous bases, they're called nitrogenous bases because the molecule has nitrogen in it. And these can be separated into two types. There's a purine nitrogenous base and there's a pyrimidine nitrogenous base. The difference between them is that the purines are double ring structures and the pyrimidines are single ring structures. And within the purine group, there are two nitrogenous bases. There is adenine, or just simply A for short, and there's guanine, or simply G for short. Within the pyrimidine group, these are single ring structures, and there is cytosine, thymine, and uracil within that group. And the way I remember the ones that are in the pyrimidines is that it spells the word cut, C-U-T. Of the sugars that are part of nucleic acids, it's one of two sugars. And you'll remember this from our carbohydrate discussion from last lecture. There's either ribose or deoxyribose. And these two sugars are identical with the exception of one component. On the second carbon of deoxyribose, it's missing a hydroxyl group or an OH group. And that's why it's called deoxyribose. So there's one less oxygen. And then another part of this is that you get a phosphate. There's a phosphate group involved in each nucleotide. A little bit of nomenclature. When a nitrogenous base attaches to a sugar, that nitrogenous base plus sugar, so for example, adenine with ribose sugar, once it attaches to the sugar, it changes its name from adenine to adenosine. And I just remember that it's the S-I-N-E at the end because there's a sugar added to it. And so if you ever hear the term adenosine, it implies that there's the nitrogenous base and the sugar attached. That'll become clear in the next slide. And so the uses of nucleic acids within biological systems, within human beings, the first place where they're used are as single nucleotides. And so these single nucleotides are involved in something referred to as energy capture and transfer. So a molecule that you've likely all heard of is ATP, or adenosine triphosphate. That adenosine, if you remember from the nomenclature, that implies that there is the adenine, nitrogenous base, plus the sugar attached. And not only is there the adenine plus the sugar, there's triphosphate, so there's 3 phosphates attached. So it has one sugar, one nitrogenous base, and phosphate groups. So this is a single nucleotide. Adenosine diphosphate is similar. Instead of having 3 phosphates, it just has 2, and also involved in energy capture and transfer. NAD and FAD are 2 molecules that, if you remember from cellular respiration, these are molecules that store high energy electrons that allow for energy transfer. And these are also examples of single nucleotides. The other time that you have single nucleotides used in biological systems is in what they refer to as cell-to-cell communication. And the molecule that is most known in cell-to-cell communication is something called cyclic AMP. or CAMP. This molecule is a nucleotide, adenosine monophosphate, and the way that it works is that normally a signal will reach the outside of a particular cell, and when that signal reaches the outside of the cell, it will cause changes on the inside of the cell to actually increase this cyclic AMP. And this cyclic AMP will go and tell certain genes to be turned on so that you can upregulate certain proteins in the cell. And so it acts as a communicator, so in cell-to-cell communication. The particulars of this, we'll get into some specific examples, mostly in the renal section, when we talk about some of the hormones that are involved in regulating salt. But for now, just know that it's an example of a single nucleotide, and this single nucleotide is involved in cell-to-cell communication. The best known place where nucleic acids are, the best known place where nucleic acids exist in living organisms is in the form of nucleotide polymers. And these nucleotide polymers are a place where there's information that is stored and transmitted. And this storage of information is in the form of DNA and RNA. And these two molecules are nucleic acid polymers, or nucleotide polymers. They're linked up from the sugar of one nucleotide links up to the phosphate of the next. And so you sort of get this backbone of sugar alternating with phosphate, sugar alternating with phosphate, sugar alternating with phosphate. And then the nitrogenous bases that are linked up with this sugar phosphate backbone, they interact with each other to make what they refer to as base pairs. And so when we look at nucleic acids, if you look at some of the nomenclature, this green area is the phosphate area, and this green molecule, so that's the phosphate. It's actually attached to the 5', the fifth carbon. So they refer to that as the 5' end because it's linked up to the 5th carbon of the sugar. And so the 5' end is the end with the free phosphate, and it's linked up, so the phosphate is linked up to the sugar, and then the phosphate is linked up to the sugar, so it goes phosphate, sugar, phosphate, sugar, phosphate, sugar. Notice that at the end of this chain of nucleic acids, there's no phosphate attached. So it's an end with a free sugar. There's no phosphate attached to that sugar, and that phosphate normally attaches to the 3rd carbon. And so they refer to that as the 3' end. And so the 3' end of a nucleic acid has a free sugar, and that's either ribose or deoxyribose. And then you can see that attached to each sugar is the nitrogenous base, which is in blue here. So there's a purine here because it's a double ring structure. Here's an example of a pyrimidine that's attached to the sugar, another purine that's attached to the sugar. So things to know, 5' end has a free phosphate. The 3' end has a free sugar without anything linked up. If we look closer at DNA, DNA uses deoxyribose sugar. DNA, so deoxyribo nucleic acid, that deoxyribo comes from the name deoxyribose. And there are 4 nitrogenous bases that exist within DNA. There's A, C, G, and T. So adenine, cytosine, guanine, and thymine. The other thing with DNA is that it forms a double helix structure. The nitrogenous base from one strand hydrogen bonds with the nitrogenous bases from the strand that is beside it. And the way that the nitrogenous bases form hydrogen bonds with each other there's only certain base pairs that can happen. Adenine in DNA will always base pair with thymine, and cytosine will always base pair with guanine. And if you look closely at the chemistry, Adenine with thymine produces 2 hydrogen bonds. Cytosine with guanine produces 3 hydrogen bonds. And we'll see a picture of this. I've tried to figure out ways to remember how many hydrogen bonds are between each base pair. And so the only thing I've been able to come up with is that AT makes the word at, which has 2 letters, and it has 2 hydrogen bonds. CG, C is the 3rd letter of the alphabet, and it makes 3 hydrogen bonds. That's how I kind of remember it in my head. It's not a great mnemonic device, but it's at least something. So the way that DNA is structured is there's that 5' end with the free phosphate, all the way to the 3' end with that free sugar. And one strand of DNA is oriented in the 5' to 3' end, and the strand that it hydrogen bonds with is actually in the 3' to 5' direction. So one strand is going in one direction, the other strand is going in the opposite direction. So it's oriented sort of in a flipped orientation. And this type of orientation is referred to as the anti-parallel orientation of DNA. So one strand is in 3' to 5', the other strand is in 5' to 3'. So there's the anti-parallel orientation. If we look at the nitrogenous bases, notice that A hydrogen bonds with T, and there are 2 hydrogen bonds between these nitrogenous bases. That dotted line stands for a hydrogen bond. If we look at the base pairing between cytosine and guanine, there are 3 hydrogen bonds between them. A and T, again, 2 hydrogen bonds. G and C, 3 hydrogen bonds. That'll never change. The number of hydrogen bonds between base pairings will never change between A and T and C and G. RNA is a bit of a different molecule. Rather than deoxyribonucleic acid, it's referred to as ribonucleic acid. And that is just because the sugar that's involved in RNA is different. We use ribose sugar instead of deoxyribose sugar. It's generally single-stranded, and it uses pretty much the same bases as DNA with one exception. So there's adenine, cytosine, guanine, and instead of thymine, it uses this nitrogenous base called uracil. The base pairing is identical as in DNA, except uracil always base pairs with adenine, and adenine always base pairs with uracil. That's the only exception. In terms of proteins, when we talk about proteins, proteins are polymers of amino acids. And if we look at the main structure of an amino acid, there's a few things that every single amino acid has in common. On the left side, you can see that there is an NH2 group attached to a carbon. That's referred to as an amino group. And on the right side here in green, you can see that there is a carboxyl group or a carboxylic acid group. And so that's where the name amino acid comes from. There's an amino group and an acid group, so amino acid. Every amino acid has a hydrogen bound to the central carbon, and this R group, this group is the one that varies between amino acids. Some amino acids are very simple and might just have a single hydrogen attached. Some might have another carbon attached. Some have a ring structure attached. But this R group is what changes between the various amino acids within proteins. Amino acids get linked up with each other via something called a peptide bond. And so if we look at 2 amino acids side by side, there's the carboxyl group of one amino acid and the amino group of the second amino acid undergo a specific type of reaction called a dehydration reaction. And this dehydration reaction causes the elimination of water. So you can see where the water comes from. It comes from the OH group of the carboxyl group and one of the hydrogens from the amino group. And you end up with water being eliminated. And the type of bond that you're left with is a carbon with a double bound oxygen, and then to that same carbon there's a nitrogen group, and to that nitrogen group there is a hydrogen. So that C double bond O, bond N, bond H, this particular bond is called a peptide bond. And you can imagine that in a protein there would be many amino acids that are bound with many peptide bonds. And that's how we get long chains of proteins. Each of those amino acids is bound with this peptide bond. 20 different amino acids occur in naturally formed proteins within the human body. Amino acids, about most of them, actually more than half of them, our body can make on its own. So we can take different molecules and we can convert those molecules into various amino acids. But there are certain amino acids that our body can't make. We have to get them from the diet because we just don't have the enzymes to create them. Those types of amino acids are referred to as essential amino acids. I'm not going to make you memorize the list of essential amino acids, but just know that there are certain ones that our body cannot make. So if you ever hear the term essential amino acid, know that that is one that must be obtained from the diet. When we talk about protein structure, there are 4 levels of protein structure. There's the primary structure of protein, which is the simplest way to describe a protein, and that's simply just the sequence of amino acids. So I'm not going to expect you to memorize all the amino acids, but an example of the sequence of amino acids would be aspartate, arginine, glycine. And this would continue on in large proteins, and it would just be the list of amino acids in order that they occur. But we know that proteins don't exist in general. They don't exist in simply just strands of amino acids. There are multiple more complex structures and ways that these amino acids interact with each other. And so the secondary structure of protein is once you have all those amino acids linked up with each other, those amino acids will form these things called alpha helices. So these spiraled shape of chains of amino acids. It's called an alpha helix. Or they produce these things called beta pleated sheets. And these sheets of amino acids will lay on top of each other and interact with each other to make proteins. So while there is a strand of amino acids, they sort of make this helix structure. But proteins get a little bit more complicated in their structure, and there's something called a tertiary structure of proteins, whereby these alpha helices will fold in on themselves to create a protein subunit. So you can see that there's all of these alpha helices, and they're folding in on each other, and it makes this more compact protein that is referred to as a protein subunit. The fourth structure of protein is something called the quaternary structure of protein, where multiple of these subunits combine together to produce a fully functional protein. And so in most cases, you get these things called globular proteins. So you can see, here's an example of hemoglobin, and you can see that there are 4 protein subunits. There's 2 of these blue protein subunits, and there's 2 of these purple protein subunits that come together to make a functional hemoglobin molecule. There's also the type of protein that exists as long fibrous strands, and many of these long fibrous strands interact and twist around each other to make these long fibrous filamentous proteins. And this would be found in something like collagen, which is a connective tissue within your body. So you can either get these globular structures with the quaternary structure of proteins, or you get these long fibrous proteins. And so we'll talk mostly about the globular proteins. We're going to go into depth on hemoglobin when we talk about the respiratory physiology. So stay tuned for that. When we talk about functional proteins, proteins can be a number of things. They can be enzymes, so things that help chemical reactions in the body occur. They can be transporter proteins, so there can be some sort of a protein that is lodged within the cell membrane that allows for certain substances to enter and leave the cell. Proteins can exist as signaling molecules to allow for certain genes to be turned on and off within a particular cell. They can be used as receptors. So different hormones will attach to certain receptors on cells. Proteins are used in the body as binding proteins. So the example of hemoglobin, it exists within red blood cells and it binds to oxygen to allow for transport of oxygen from one part of the body to the next. Proteins are also used in the immune system and the immune response as immunoglobulins or antibodies. So those are just proteins that can help identify foreign substances within the body. And proteins are also used as for regulatory purposes, with the turning on and off genes within the nucleus of the cell, and allowing for certain genes to be upregulated or downregulated depending on different signals that that the cell will get. A little bit on protein binding. There are a couple of terms you should know. The first is a term called ligand. So a ligand on a protein is a molecule that attaches to the protein. So the ligand is the molecular ion that actually binds to a particular protein. The term binding site is the specific location on a protein where a ligand attaches to. There is something within proteins called protein specificity, and it really depends on the specific protein. Some proteins allow for attachment of many different types of ligands, and some proteins only allow for the attachment of one particular ligand. We'll talk about many examples of proteins in the upcoming lecture. When a ligand actually attaches to the protein, you may have heard of the lock and key theory of protein ligand interaction, and that's a fine model to use. But one of the things that you should recognize is that a ligand attaching to a protein, it doesn't have to be an exact match. It's a near close match. It's something called the induced fit model, whereby as a ligand approaches a protein, the chemical forces between the protein and the ligand itself will produce hydrogen bonds, will cause some van der Waals forces to be created between the two molecules, and it will somewhat change the binding site when the ligand is in close proximity to allow for the ligand to attach properly. There are certain factors that affect protein binding. Most proteins in the body have some sort of an inactivated form. And the reason for that is because a lot of proteins in the body will be produced in one location and will be transported to another location. And it's not until they're transported to the right location that these proteins should be activated. And so this thing called proteolytic activation is that a protein is inactive until certain components of that protein are removed. This is very true for enzymes. Specifically, one of the enzymes that is important to remain inactive until it's in the right location is a protein enzyme called trypsinogen. And trypsinogen we'll learn more about it when we talk about our GI section, but trypsinogen is a protein within the body that acts as an enzyme to break down proteins. And so this is very important in digestion. So when you ingest proteins in your diet, trypsinogen will act to break down those proteins into single amino acids. And it's exceptionally important for this trypsinogen to stay inactive until it gets to the gastrointestinal tract that has a protective layer that doesn't allow it to interact with your own body cells. Because part of your own body cells are proteins. And so if this enzyme becomes activated too early, and in a location that it shouldn't be activated, you're going to start breaking down your own cells. So this is an example of a protein that is produced. It's produced in the pancreas, and when it's released, it goes from the pancreas and it goes into the small intestine. And the small intestine has a layer of mucus that lines it, so that once it's in the small intestine, it doesn't interact with the cells of the small intestine because the mucus protects it. So once it's in the small intestine, that's when trypsinogen can actually be activated into trypsin to start breaking down proteins. If it's activated too early, it's going to start breaking down your own cells. So we're going to talk about this in a lot more detail in the GI section. And I'll also talk about, within the GI section, what happens when this gets activated too early. And there are some disease processes that result from trypsinogen being activated too early. A little bit of nomenclature. Often when you have an inactive protein or an inactive enzyme, that inactive enzyme has the suffix ogen. So anytime you see the suffix ogen on anything, it means that it's likely an enzyme that is in inactive form. So the example, trypsin ogen. In inactive form, it has that ogen attached. When it becomes activated, that suffix ogen goes away and it just becomes trypsin. This is one of the questions that I like to ask on the exam. I'll usually throw some enzyme names and I'll say which one is in inactive form, and you typically just have to look for the one that has ogen at the end of it, and that's the answer. So an easy question for if you recognize the nomenclature. And I'll throw enzymes that you've never heard of, but just look for the one that has ogen, and that's the inactive one. Other factors that affect protein binding is something called allosterism. An allosteric activator, or allosterism within a protein, is a substance that will bind to the protein, but it binds at a site that's far away from the actual binding site. And once that substance binds to the protein, it will then cause the protein to change shape, and it will allow a ligand to bind. So an allosteric activator causes a protein to change shape, allowing for a ligand to bind. Hemoglobin is a great example of an allosteric protein. When oxygen binds to the actual molecule of hemoglobin, or to one of the globin, to one of the heme aspects of hemoglobin, hemoglobin will change its shape to allow for more oxygen to bind easier. This will be discussed in more depth within the respiratory section of the course, but hemoglobin is a very good example of a protein that exhibits allosterism. Cofactors also affect protein binding. Cofactors are substances that are required for an active binding site. So in certain proteins, a cofactor will have to bind to an inactive protein, and then the ligand actually binds onto the cofactor in order to activate the, actually activate the protein. Without the cofactor present, the ligand can't bind to the protein. Once the protein is activated by the cofactor and the ligand's binding, the protein will be able to do its job. And at the end of it, at the end of this protein doing its job, the cofactor remains unchanged. And the cofactor will release from the active protein and can then go and activate another similar type protein. An example of where cofactors are very much required are within the blood clotting proteins. Without certain cofactors, blood clotting cannot happen. So one question is, how are allosteric modulators and cofactors different? So the main answer to this is that an allosteric modulator does not attach to the actual binding site of the protein. it attaches to a site away from the protein binding site to allow for the binding site to become activated. Cofactors, on the other hand, actually bind to the binding site of a protein. So one attaches away from the binding site, the other attaches right to the binding site. The last type of way that proteins get activated or sometimes inactivated are through things called covalent modulators. These are generally permanent changes within a protein. And covalent modulators are atoms that bind to a protein to either activate it permanently or deactivate it permanently. The most common and well-known covalent modulator is phosphate. Adding a phosphate group to a protein is something called phosphorylation. And one of the best known covalent modulators is penicillin. It helps, it actually binds to proteins within bacteria and permanently inhibit them from working. And without these proteins within the bacteria cannot synthesize the essential components of the cell wall, and the bacteria end up bursting and dying. And so that's generally how penicillin will work. It covalently modulates the proteins within a bacterial cell wall. We'll talk a little bit about body compartmentalization. And this compartmentalization exists within cells. It exists within body tissues, between organ systems. There are many levels of compartmentalization. Having compartments within the body has some advantages. The main advantages are that we can separate biochemical processes that might conflict with each other. Certain parts of the body might want to build up glucose. Other parts of the body want to break it down to use for energy. If you didn't have those processes separated from each other, you would end up having a situation where you're building up glucose and breaking it down at the same time, and there's no net effect. So it just allows the contents of one part of the body to differ significantly from another part of the body. The main disadvantage of having compartments is that sometimes it's hard to move things from one part of the body to another, and this is where a lot of the physiologic and cell biology processes take place, and they come into play to try to move things from one part of the body to the next. If we look at, this is a sagittal view of the human body, and you can actually see that there's compartmentalization within the body cavities. You can see that there's the cranial cavity and the spinal cavity. So there's where the brain and the spinal cord are completely separate from other parts of the body, like the thoracic cavity. The thoracic cavity that contains the lungs and the heart are completely separate from the cranial cavity, and they're actually completely separate from the abdominal cavity. So there's this compartmentalization, and each one of these compartments has organs within it that serve a very different purpose. Even within the thoracic cavity, you can see that there's a compartment within a compartment. So the place where the heart would lie within the pericardial sac, that is completely separate from where the lungs are within the pleural sac. And the heart and lungs, while they work together in most circumstances, and they should work together, they have very different functions. The heart is used to pump blood to parts of the body. The lungs are used to oxygenate blood and to get rid of carbon dioxide. Very different functions, and so they need to be separated through compartments. There's also fluid compartments within the body. Each cell is separated from each other, and there's fluid within the cell. So this fluid compartment is called the intracellular compartment. That's everything inside the cell. And then there's a bunch of fluid on the outside of all the cells, which is referred to as the extracellular component of the body. And so these fluid components are separated, and the actual composition of what's within those fluids is vastly different. And that's what makes a lot of the ability of cells to communicate with each other. That's what makes it functional. That's what allows for different biochemical processes to happen, because the constituents in each of the intracellular component and the extracellular component are so different. If we look beyond just the intracellular and extracellular components, the actual extracellular fluid has two components. There's, within the extracellular component, outside of the cells, there is the fluid component within the blood, and that's referred to as the plasma. That's the fluid component of blood. There's no cells in that. That's on the outside of the cells of the blood, so it's extracellular fluid. And then there's another type of fluid within the extracellular component, which is called the interstitial fluid. This interstitial fluid is really the fluid that is surrounding each cell. And so that makes up the extracellular fluid compartment. So while you have two fluid compartments, there's the extracellular and intracellular, your extracellular is separated into two further fluid compartments. The fluid between cells, and then the fluid that is the component of blood. We'll talk more in depth about that as well. Most compartments of the body are separated by membranes. So we can actually look at the, say, the pericardial compartment. Well, there's surrounding the pericardial, or surrounding the heart itself, is this thing called the pericardial membrane. And so this separates the sac that the heart is in from the sac that the lungs are in. And so this pericardial sac, if we look very closely at this pericardial sac, we can actually see that there are cells that line this pericardial sac. And if we look at those cells, we can see that each cell is separated from each other by a membrane. So there's sort of a compartment within a compartment. And so each individual cell has a cellular membrane that separates them. So while we are separating the heart from the lungs, we're also separating each individual cell within the pericardial sac from each other by a membrane. And the cell membrane is a really important part of the compartmentalization process within physiology. And within animal cells, this cellular membrane is comprised of phospholipids. And it's specifically a double layer of phospholipids. They refer to it as a phospholipid bilayer. So the function of the cell membrane is really to create a physical barrier that separates the intracellular fluid compartment from the extracellular fluid compartment. It acts to regulate ionic and molecular exchange within the external and internal environment. It acts to communicate with other cells, and it plays an important role in structural support of the cell. The cell membrane is comprised mostly of lipids and proteins, but there are a few carbohydrates that are linked in as well. The lipid component is by far the most prominent of the cell membrane, and it is mainly phospholipids. So if we look at how the phospholipids are arranged within a cell membrane, they are arranged in a bilayer. And so phospholipids, if you remember, there's a glycerol backbone, there are two fatty acid chains, and there is a single phosphate attached. And the way that they arrange themselves is that the phosphate group tends to be hydrophilic, meaning that it prefers to be in contact with water. And this is referred to as also being polar. So this is the phosphate is a polar molecule. And these polar head regions of the phospholipid bilayer prefer to be in touch with the water component, either on the inside of the cell or the water component on the outside of the cell. So you can see that the phospholipid heads are always in contact with some sort of a water component. And the way that they're arranged is that the phospholipid tails, these tails are hydrophobic or non-polar, and they don't like to mix with water. So these hydrophobic tails tend to be on the inside of the cell membrane, so that you always have a hydrophobic component between the layers of phospholipids. And so this makes a bilayer, and it ensures that only the polar parts of the phospholipid are in contact with water. So the major model of the cellular membrane is something called the fluid mosaic model. And what this implies is that the cell membrane is not a rigid or a static structure. The phospholipid bilayer or the cell membrane is always constantly changing, and proteins can be removed and inserted without disrupting the overall integrity. So proteins tend to change locations, and it's a very fluid membrane. That's why it's called the fluid mosaic model. One of the things that helps with some structural integrity within the cell membrane so that it doesn't fall apart is the incorporation of cholesterol molecules. And if you remember from the lipid section within lecture 1, these cholesterol molecules are lipid-like molecules, and you can see that they're inserted in and around the non-polar tails. And the more cholesterol you have within your cell membrane, the more rigid it is. But it helps maintain structural stability. So cholesterol isn't all that bad in moderation. So not only does it allow for the structural stability, but it also helps the membrane stay quite flexible over a wide range of temperatures. It has a higher freezing point, so it allows for the membrane to stay fairly fluid. The carbohydrate component within the cell membrane, there tends to only be carbohydrates on the external surface of the cell membrane. It's not very common to have a lot of carbohydrates. The best known example of carbohydrates on the external surface of the cell membrane are the carbohydrates that help determine blood type. So the ABO blood types are determined by various carbohydrates that are attached to the external surface of red blood cells. So these carbohydrates help your body recognize that your cells actually belong to you. So this is one of the reasons why if somebody were to get a blood transfusion, if you give them the wrong type of blood, well, the body will recognize those carbohydrates on the outside of the red blood cell, and your immune cells are going to destroy those cells because they recognize that the carbohydrate isn't correct. And so that's one of the reasons why it's exceptionally important to give someone the proper blood type. And it's all based on the carbohydrates. So that's the best known example of carbohydrates on the cell membrane. If we look further into the cells, what are cells divided into? Well, there's more compartments. So most cells within the body are divided internally within the cytoplasm component and the nucleus component. The fluid component of the cytoplasm is called the cytosol, and the structures within the cytosol are called organelles. And these organelles are separated, most organelles are separated from each other by more membranes. So there's compartments within compartments within compartments, and it just depends on the scale that you're looking at. One of the organelle components of the inside of the cell are these things called the cytoskeleton components. Many things make up the cytoskeleton components. There's microvilli, there's microfilaments, there's microtubules, there's intermediate filaments. This sort of acts as the scaffolding of the cell. It helps stabilize the cell. It helps stabilize various organelles within a certain position within the cell. And it also serves as a transport function. that some of these organelles can act as railways, a railway track to allow for various molecules to move throughout the cell to go to various organelles. So cytoskeleton component is an important part of keeping the integrity of the cell, stabilizing the position of the organelles, and also acts as a transporter within the cell. Some specific organelles that are membrane bound, that are separated from the cytosol, are things like the mitochondria, referred to as the powerhouse of the cell. This particular organelle, it's a double membrane organelle, and it serves the purpose of participating in cellular respiration processes. So the citric acid cycle, or the Krebs cycle, that we'll talk about in depth, occurs within the mitochondria, within the fluid of the mitochondria. And the other aspect of cellular respiration that occurs within the mitochondria is the electron transport chain, which we will talk about in depth. It occurs within the intermembrane space of the mitochondria. The big thing here is that this particular organelle is completely separate from the rest of the cell by membranes. So you can imagine that some of the processes that are happening within this organelle are completely different than the processes that are happening within the fluid component of the cell. Just again, serving the purpose of highlighting the importance of compartmentalization. The endoplasmic reticulum is another organelle within the cell that is membrane bound. There's really two types of endoplasmic reticulum. There's a rough endoplasmic reticulum. You can see this sort of stippled appearance to the rough endoplasmic reticulum. This stippling is created by the presence of ribosomes. Ribosomes are a small site within the cell of protein synthesis. So when proteins need to be made, they're made on ribosomes. And this site of protein synthesis, the proteins will be made, and they will be brought into the endoplasmic reticulum to further be processed. There's also a component of the endoplasmic reticulum that is the smooth endoplasmic reticulum, and the only difference between this, the actual structure of it, is that there are no ribosomes on the smooth endoplasmic reticulum. There is a functional difference, though. The main functional difference between the rough and the smooth endoplasmic reticulum is that the smooth endoplasmic reticulum is the site of lipid synthesis and is also the site of calcium storage. And we'll get into that in our cardiovascular section. The Golgi apparatus is often very closely related to where the endoplasmic reticulum is, and it sort of is arranged as this sort of stack of pancakes kind of structure. And the thing that the Golgi apparatus does is it takes proteins and it packages them for transport through the cell, and can be then brought out of the cell and be transported to different areas within the tissue. There are some other cellular contents. There's peroxisomes. Peroxisomes help with fat metabolism. They help degrade some of the long fatty acid chains for use within the cell. Lysosomes are a little bit bigger than peroxisomes, and these lysosomes are really the digestive component of the cell. They have very powerful enzymes within them, and it's extremely important that they are separate from the entire cell, because if these lysosomes, if they weren't, the enzymes within the lysosomes would actually kill the entire cell. So again, highlighting the importance of compartmentalization within the cell. Large component within the compartment within the cell is the nucleus. It's often referred to as the brain of the cell. It contains the DNA. There is a separation between the cytosol and the nucleus, and this separation is a double membrane structure, and it's called the nuclear envelope. The nuclear envelope. Within the nucleus, there are also little compartments within there called the nucleoli, and these nucleoli are where RNA is synthesized. So an example of how compartmentalization allows for the separation of function within the cell. You don't need to memorize this, but this is just showing you that this is within a cell, and so here's the nucleus. The yellow is the cytosol. The bluish color, the light sort of purple-ish color, is the endoplasmic reticulum, and then this stacked-like structure are the Golgi. And so if we look at how different things are compartmentalized, again, don't memorize this, just a way to demonstrate compartmentalization. Within the nucleus of the cell, this is where the DNA is. This is also the site of where RNA is going to be synthesized. Once the RNA is synthesized, it is then taken out of the nucleus and brought into the cytosol. So once it's brought into the cytosol, the mRNA can actually attach to ribosomes, and that mRNA can be translated into various proteins. And all of those various proteins have different fates. Some of those proteins are just remain as free proteins within the cytosol. Some of those proteins are targeted for various organelles, and they go into the specific organelles. Some of those proteins are actually packaged within the endoplasmic reticulum and then further transported through the Golgi to be brought outside of the cell. So it really depends on what component you're looking at, but you can see that the function of making mRNA is very separate from the process of making proteins. And it's very different in the process of making proteins that are going to be used within the cell. It's within a different compartment than the proteins that are going to be made for use outside of the cell. So everything has its own little place within the cell. That's really what this is talking about. We'll stop there for today, and we will continue on this sort of pre-physiology information next class, and we will get into a little bit of energy metabolism. And so if you have any questions, you can always email me, or you can use the chat function within on the website, and I'm happy to answer any questions you have.